I will introduce our second speaker, Dr. Sandra Martinez Martin. Hope I pronounce your last name correct. So Sandra did all her study in Spain. Now you also see it on the screen, of course. Graduated in health biology from Universidad de Alcalá in 2014, and followed up by a master's degree in biomedicine from the University Autónoma de Madrid. And then she continued to do a PhD in Barcelona, where she already started to working on this very interesting project that she will tell us more about tomorrow. It's a first, basically in class or in modality MYC inhibitor that is going to be very powerful in treating some of the oncology indications. With that, I'll hand over to Sandra.
Thank you. Thank you very much, Johan, for the introduction. Good afternoon, everyone. With today's talk, I would want to tell you about Peptomyc's journey in the design and the implementation of a biomarker discovery program, in which we also meant to establish a pharmacokinetic and pharmacodynamic relationship for OMO-103 as we approached the clinical setting. A very brief introduction about myself. I'm originally from Madrid, and I moved to Barcelona, as Johan mentioned, for the PhD studies. I worked in the laboratory of Dr. Laura Socek at the Valdebrán Institute of Oncology, and during my PhD studies, Laura and a postdoc in her lab, Dr. Marie-Ève Beaulieu, founded Peptomyc, a spin-off biotech company that is aimed at bringing OMO-MYC, the best direct MYC inhibitor known to date, to cancer patients. After my PhD, I joined Peptomyc as a postdoc, and with the idea of leading this biomarkers project. So in here, just a picture of the current research team, as well as some media releases that have accompanied our project up until now. So Peptomyc's goal is to offer new and hopefully better cancer treatments. Some recent statistics show that over 17 million cancer cases will be diagnosed yearly, and more than 10 million people will die annually of this dismal disease. As per the current therapies available, they all have in common, or they all share some challenges, such as the development of acquired resistance, lack of efficacy, and some toxicity. But this actually leaves room for the development of new therapeutics that can overcome these hurdles. And this is where targets that have been longly considered undruggable come into play, like MYC. MYC is an oncoprotein and a transcription factor that appears deregulated in up to 70% of human cancers, and it is required for tumor development and survival. However, to date, there is no direct MYC inhibitor that has been approved for clinical use. So what's the solution that we propose here? We propose the use of OMO-MYC. OMO-MYC is a mini protein. It's actually based on the BHLH Lucent super of MYC itself. It was designed by Laura Socek more than 20 years ago, and she introduced 4 amino acid substitutions that changed the dimerization specificity, allowing it to form both homodimers as well as heterodimers with both MYC and MAX, which is MYC's obligate partner, and it's required to bind DNA and induce the transcription of MYC target genes. So OMO-MYC basically competes with MYC-MAX transcriptionally active dimers in its 3 dimeric forms, and it blocks MYC-dependent transcription, eventually leading to the blocking of cell proliferation. In 2019, a study led by Marie-Ève Beaulieu showed that the recombinantly produced OMO-MYC mini protein had cell-penetrating properties, and this meant that OMO-MYC could become a drug. It could be used for treatment. In this same study, we actually showed that it was effective against multiple tumor types in vitro, and it showed therapeutic efficacy in mouse cancer lung cancer models upon both intranasal and intravenous administrations. So all this work eventually led to the clinical setting. And after completing all the mandatory preclinical safety and tox studies, OMO-103, which is OMO-MYC under its clinical product name, was granted approval to be tested in a first-in-human clinical trial, whose primary objective was to evaluate the safety and tolerability in all comers, that is, patients with advanced solid tumors of different kinds. And then as secondary objectives, we included the pharmacokinetic analysis, the evaluation of preliminary efficacy, the establishment of a recommended phase 2 dose, as well as the assessment of anti-drug antibodies against OMO-103. And additionally, as exploratory objectives, we plan to evaluate the predictive and pharmacodynamic biomarkers, as well as MYC levels. So this first-in-human phase 1 clinical trial was divided into 2 stages. The first stage that was the accelerated titration, who included 2 patients, each corresponding to a different cohort, that were treated with a dose that was below the expected pharmacologically active dose, followed by a dose escalation that goes under the traditional 3+3 design. So we had up to 4 dose levels, the extra dose levels up to dose level 6. And finally, dose level 5 was chosen to be the recommended phase 2 dose. So in this study, we included 22 patients. All had been heavily pretreated and then received intravenous infusions of OMO-103 once a week. So the study was a complete success because the drug demonstrated safety. And we also show some promising preliminary efficacy results that we think are worth mentioning, especially 8 out of the 12 patients that reached the CT scan at 9 weeks showed their disease stabilized, and even 3 more cases that I would like to mention in specific are a pancreatic ductal adenocarcinoma patient and a sarcoma patient who stayed for a little bit over 6 months in the study, and a salivary gland adenocarcinoma patient who stayed for a little bit over 2 years. So these 3 cases are special for us because these show long-lasting, stable diseases. So this was a complete success for us. So this is a little bit in a nutshell the journey that has brought us to a clinical to the clinical setting. But now I would like to tell you a little bit more about the biomarkers and why were they important for our project. Very briefly, biomarkers are measurable characteristics of a therapeutic response or a normal or pathological process that can be objectively and reproducibly assessed. And in our case, for OMO-MYC and OMO-103, we were particularly interested in the discovery of pharmacodynamic biomarkers that would allow us to understand what response we are inducing in the individuals that we treat. Like I mentioned, there is no direct MYC inhibitor that is clinically approved, and that means that there are also no biomarkers described for these type of molecules, and special for OMO-MYC because it has a very unique mechanism of action. So we came up with a plethora of experiments to understand at the preclinical level. And one other key aspect of our project was the establishment of a pharmacokinetic and pharmacodynamic relationship. So we wanted to understand the link between the exposure to OMO-MYC and the response that we observe. It is important to note that OMO-MYC is a biologic, which means that we really want to know how much of the full-length mini-protein is reaching our target tissue. And at the time, we could analyze full-length OMO-MYC in mouse serum and in human serum by radioimmunoassay and electrochemiluminescence assay, respectively. But for tissues, we were limited with the use of immunoblot and immunofluorescence. And these 2 techniques have their own intrinsic limitations, mainly the fact that these are semi-quantitative. So we also had performed an experiment with radio-labeled OMO-MYC that hinted at a different PK profile between serum and tissue. So we wondered whether, with the use of targeted proteomics and the help of Biognosys, we could figure out whether by mass spec we could quantify OMO-MYC in our samples. We ran a first pilot experiment in which we used 180 tumors coming from xenograft mouse models that had been treated with 2 different doses of OMO-MYC and for 2 treatment durations. Then we collected the fresh frozen tumors and ran proteomics with the help of Biognosys. In this case, we chose two stable isotope standard peptides that were unique to OMO-MYC sequence. So these are the ones that correspond to the sequence in which we introduced the 4 amino acid substitutions. And the results showed that we could detect OMO-MYC in every tissue that we analyzed. And actually, and somewhat expected, there was dose dependency. So we could quantify higher levels of OMO-MYC in those mice that have been treated with the highest dose, 75 milligrams per kilo, compared to those that were treated with 50 milligrams per kilo. So we jumped into a second experiment, and we wanted to confirm this difference in the PK profile between the two compartments. So in this case, we inoculated tumor cells, allowed them to grow up to 400 cubic millimeters, and randomized the mice in 5 groups. All of the mice were treated with one single intravenous dose of 50 milligrams per kilo of OMO-MYC, and then were euthanized at different time points, 5 minutes, 2, 24, 48, and 72 hours. And then we collected both serum and tissue in 2 forms. Fresh frozen to be able to compare with the previous experiment, and FFP, because this would be the only kind of sample that we could get from our biopsies from the patients of the clinical trial. And like I mentioned, OMO-MYC is a biologic, so in order to increase the coverage, because we want to be as close to full-length OMO-MYC as possible, additionally to the 2 unique sequences, we included 2 more sequences that are actually shared with the MYC protein, as well as a negative control that would allow, or that would ensure that our detection is distinguishing correctly OMO-MYC from endogenous MYC. And not only were we excited for confirming that there's actually this difference in the PK profile between the 2 compartments, as you can see, with all 4 peptides analyzed, there's increased residency in the tissue compared to serum, but also because the difference between FFP and fresh frozen was not that big, which meant that we could apply these to our clinical samples. So just to wrap up with the preclinical part, OMO-MYC is asymmetrically distributed between serum and tissue, where it accumulates with dose dependency, and it lasts longer. So now we'll be switching gears to the more clinical results. The first in-human clinical trial started in May 2021, and it overlapped partially with the preclinical experiments that we just discussed. So the idea here was to generate data with more or less the same type of techniques so that we could integrate and validate the data both at the preclinical and clinical level. One limitation that we faced with the biopsies, with the patient samples in this case, and especially with the biopsies, was how limited they are. So to run transcriptomics, we, instead of running bulk RNA sequencing that would have required a lot more of material, we chose to use spatial transcriptomics. That with just one cut, we could analyze up to 18,000 genes. And then to be able to quantify OMO-103 in these biopsies from our patients, we used stained slides that had been used for immunohistochemical analysis, so that Biognosys could extract a protein from them, and we could just have an absolute quantification of OMO-103. So very quickly, about the transcriptomic data, we analyzed paired biopsies that were taken pre and post treatment. We chose to analyze 4 to 5 regions of interest that were segmented into 2 different compartments: pan-cytokeratin positive, corresponding to tumor cells, and pan-cytokeratin negative, corresponding to the tumor microenvironment. And the results showed a transcriptional shutdown of MYC-related gene sets. So with this, we confirmed that there's target engagement in our tumor biopsies. What was very interesting here is that, well, you can see the blue dots are showing the downregulation here. So we have 2 blocks. In one column, 3 patients corresponding to dose level 3, and in the other column, 3 patients corresponding to dose level 5. So the shutdown of MYC targets was more profound in patients that were treated with a higher dose. So we were hinted at some sort of dose dependency. But when we look at the response of the patients, if we compare patients who had their disease stabilized versus those that did not, we also see a more profound shutdown in patients that responded to our treatment. So at this point, we have confirmed target engagement of our compound, but is it reaching the biopsy? Now we have the tool. So once again, with the help of Biognosys, we analyzed these biopsies, and we actually were very happy to see that in every biopsy analyzed, we could detect OMO-103 with at least 2 out of the 4 peptides that we used. The N is too small to make any conclusions about any dose dependency in terms of accumulation. And when we compare stable disease patients versus progressive disease patients, there's no statistical difference in the levels that we detect. However, it was really nice for us to see that the 2 highest concentrations that we detected corresponded to stable disease patients. So to wrap up also with the clinical part, OMO-103 shuts down MYC transcriptional programs in patient biopsies, where it promises to have long residency too, and this could eventually result in the reduction of the dosing frequency. So just before finishing up, I would like to show one of the patients that were a successful case, in our opinion, in terms of efficacy. This is a pancreatic ductal adenocarcinoma patient who stayed for a little bit over almost 7 months on treatment with his disease stabilized. So this patient, by RECIST, which is the more standard clinical criteria used to determine the response radiologically, had an 8% reduction. However, when we looked by radiomics to perform a full volumetric analysis, taking into account 3D structure and all the lesions instead of just 2D and 5 target lesions, we actually quantify a 49% reduction in the tumor of this patient. That was accompanied by an 83% reduction in the mutant variants, gene variants by ctDNA. And this case is also interesting from the biopsy point of view and from the accumulation of OMO-103, because first of all, this patient was treated with 2 different doses. So he started on dose level 3 up to cycle 4, more or less, and then was internally escalated to dose level 4, as clearly shown by the serum PK profile. But we were lucky enough to be able to take an end of treatment biopsy from this patient. The sample was collected actually 19 days after he had received his last infusion, and when we looked at it by proteomics, we could still detect OMO-MYC there, confirming the long residency that we had hinted at before. And then just some immunohistochemical analysis in here showing or supporting a little bit the case. So there's a slight reduction in proliferative cells as shown by Ka67, and a slight increase in apoptotic cells as marked by cleaved caspase 3. So just to finish up, I hope I showed enough evidence to convince you that OMO-MYC has a different PK profile between serum and tissue, where it accumulates for a longer time. OMO-103 is, to our knowledge, the most advanced MYC inhibitor, direct MYC inhibitor in development, as it has finished a first-in-human clinical trial successfully showing safety and tolerability. OMO-103 shuts down MYC transcription, and it accumulates it shows long-lasting residency in the patient biopsies, as we hinted at preclinically. And lastly, as per our future studies or directions, we started last fall a phase 1b clinical trial combining OMO-103 with the standard of care gemabraxane in pancreatic ductal adenocarcinoma patients, in which we expect to continue with the demonstration of or validation of target engagement and pharmacodynamic markers identified in the phase 1a, as well as to continue with the PK profile studies. So last, I would like to acknowledge everyone in Peptomyc's team, especially Laura and Marie-Ève, for their mentorship, the colleagues that have been directly involved in this project, Silvia, Sergio, and Laia, as well as the academic research team from Laura Saucek, especially Trini Core, who actually partnered up with me with the collaboration with Biognosys from the very beginning. Then all the rest of the bio teammates, all of our collaborators in special Biognosys for the opportunity of presenting here today. and our funding agencies. And last but not least, I would like to thank you all for your attention.
Maybe I can ask a question here. So I think it's a great combination, right, of preclinical models and, of course, first-in-human trials. You've showed quite a few CDX model there, right? How do you see the translatability of those biomarkers, from those models, into real, of course, in human, right, where it actually matters?
Yeah, so actually I think I was discussing this with Jonathan before. It is tough because we are targeting a transcription factor here. So the cellular context and each model, it's unique. Each one is different, and so are patients. So the translatability, I think we are trying to get there. I don't think we have just found yet the best way of finding these biomarkers, but we hope we manage to do so. That's why we keep on doing some more experiments and hopefully find a way.
Great, thanks a lot.